like I said, protein synthesis happens in two parts. Part 1 is transcription. Part 2 is translation. Today, the goal is to get y'all kind of in the weeds of what transcription is, be able to list the steps of transcription and what happens in each step. So the key players, what happens, what the outcomes are, all of that fun stuff. Before we get started, do we have any questions? All right, another clicky thing. So, starting us off, we have a new enzyme in the mix. It is called RNA polymerase. RNA polymerase is actually really stinking cool because it does a couple of jobs all at once. So RNA polymerase will pry the two strands of DNA apart. So it does what helicase does. It breaks those hydrogen bonds. And then it joins RNA nucleotides to the complementary side of the DNA template. So it can also only assemble nucleotides in the 5' to 3' direction. So it will open up a DNA, find the one that is going 3' to 5', build 5' to 3'. It also does not need primers, so it can just go. It has other mechanisms to tell it where to start. So RNA polymerase does essentially what DNA helicase, DNA polymerase, and primase do all in one. Very efficient RNA polymerase. And there are a couple RNA polymerases, but this is just like the general outline of what an RNA polymerase does. This is what it looks like, which is super fun and cool. We've got all of those alpha helices and beta sheets, which we'll talk about hopefully on Monday. Hopefully. But this is like a 3D model of what RNA polymerase would look like. So yeah, pries the DNA strand apart, adds RNA nucleotides to the template DNA strand, can assemble nucleotides in the 5' to 3' direction, and only the 5' to 3' direction, and does not need a primer. So, very versatile enzyme. So, that's one of the first molecular components to transcription. We also have the different types of RNA polymerase. In bacteria, there is only one. So bacteria only has one type of RNA polymerase, but in eukaryotes, we have at least 3. While there are at least 3, the only one we need or like the biggest one that we are going to talk about is RNA polymerase 2, which does pre-mRNA synthesis. So normally in like your intro bio, when we talk about transcription translation, we say the RNA polymerase comes through, makes the mRNA goes off. There's another step in there that we're going to talk about today. So the molecule that RNA polymerase actually makes is not mRNA, but it is pre-mRNA. The other RNA polymerases transcribe molecules that aren't translated. So it would be like our tRNAs or rRNAs. They make RNAs that aren't mRNA, essentially. So what we are focused on is RNA polymerase 2. Now, throughout this presentation and through most presentations, we won't necessarily focus a lot about the nuanced differences between the bacterial and eukaryotic RNA polymerases. So the factors of transcription are going to be common to both. So the way RNA polymerase 2 functions in pre-mRNA synthesis is very similar, if not identical, to the process that the one type of RNA polymerase in bacteria works. So the process of transcription is the same, but what happens afterwards is what makes eukaryotic and prokaryotic transcription different. And we'll talk about that. So as of right now, we are doing like a generalization of like, hey, this is what transcription is in bacteria and eukaryotes. Just know that eukaryotes have more than one type of RNA polymerase. So it is RNA polymerase 2 in eukaryotes that does all the transcription stuff for protein synthesis. Questions here? So, all right, woo. We also have new molecular components called promoters and terminators. Promoters are specific sequences along the DNA where transcription begins. So they are specific DNA sequences that say, hey, transcription starts here. Promoters are in both bacteria and eukaryotes. Terminators, however, are only in bacteria. Terminators are sequences that tell transcription where to stop. So they'll hit a certain sequence, the RNA polymerase will be like, oh, I'm done, and then it'll dissociate. Eukaryotes have a different process, which I will talk about. So promoters are universal; they tell transcription where to start. Terminators in bacteria tell transcription to stop. Right. So, some more terminology for you. In genetics, we use the phrases upstream and downstream a lot. So the direction of transcription, so that 5' to 3', is called downstream. So if you are moving in the direction of transcription, you are moving downstream. If you are moving in the other direction, you are moving upstream. So for example, the promoter is upstream from the terminator because it happens before the terminator does. So if we are reading from left to right, going in the direction that we normally read goes downstream, going backwards goes upstream. So in this image right here, it goes 5' to 3'. This right here is our point of reference. So everything is always based on perspective. So if this is our point of reference, going this way goes downstream, going this way goes upstream. So gene A is upstream from gene B. Gene B is downstream from gene A. So this is very, very common. You will hit upstream and downstream a lot. It's just a way that geneticists orient themselves on a molecule of RNA or DNA. This can be in RNA or DNA, upstream and downstream. So just keep that in mind. So another molecular component before we get into the steps of all of this stuff is the transcription unit. The stretch of DNA downstream from the promoter that is actually transcribed is called the transcription unit. So the whole thing is the gene. From promoter to terminator, that's the gene. The transcription unit is everything that is transcribed. I'll let you get this down and then we'll use this. So, if this DNA is our frame of reference, this is our 5' end, this is our 3' end. So going this way is downstream, going this way is upstream. So a gene is everything from promoter to terminator. That's our gene. Our transcription unit is everywhere that is actually transcribed. Anything that will be mRNA. So transcription will go from where our transcription unit is everything from where transcription starts to where transcription stops. So the transcription unit is downstream from the promoter. And our terminator is downstream from our transcription unit. Anybody have any questions here? If you do, just let me know. So, now, getting into the actual process of transcription, there are 3 parts. Usually, when we talk about transcription, or in the past, when you have talked about transcription, it's just been transcription. We're going to break it down because there are 3 steps to transcription. They are initiation, elongation, and termination. Start, make it, stop it, essentially. Break it down into 3 steps. And these steps will be the headings for the next 3 slides, so don't super worry about getting them down. So, transcription in 3 parts, starting with initiation. I could have made that font bigger. The start point is the actual nucleotide where RNA polymerase begins synthesis. So the first new nucleotide is the start point. The first new nucleotide is the start point. So promoters will include that start point as well as several dozen nucleotide pairs upstream from the start point. So if this is the promoter, the start point is downstream from where the promoter actually starts. There are a lot more nucleotides in the promoter than just the start point. When we talk about gene regulation, we will come back and spend a lot more time with the promoter. But for now, it is a lot bigger than just a start point. The start point is kind of towards the end of the promoter. And this is several dozen nucleotide pairs. So lots of nucleotides. So this is the location of the first step of transcription. This is where it all begins. Promoter, start point. So, in eukaryotes, transcription factors will help mediate RNA's binding to the promoter. So there are special things called transcription factors. They can be proteins, they can be chunks of RNA, they can be a lot of different stuff. Sometimes there's carbohydrates involved. Transcription factors will help RNA polymerase bind to the initiation site. The complex, so RNA polymerase 2 bound to the promoter and all of the transcription factors, so everything when it is all in place, that is called the transcription initiation complex. So when everything is all put together, it is called the transcription initiation process. And there is a special sequence, which was mentioned in that crash course video, called the Tata box, where I think the sequence is TATAAA. Right? There's a special thymine adenine sequence that is that should be a Y. That plays a very, very important role in that formation of the transcription initiation complex. That kind of helps anchor everything all together. And there's a picture on the next slide that kind of helps visualize. So, initiation is thanks to all of these things called transcription factors that will combine with the RNA polymerase 2, help it bind to the promoter, the RNA polymerase, and all of the transcription factors come together and make the transcription initiation complex. And part of that promoter is called the Tata box. Of course it's going to do that. Come on, get back. There we go. This is what I wanted to do. We're going to need to calibrate that. So, transcription factors, RNA polymerase, promoter, is called the transcription initiation complex. So, this is kind of what it looks like. We have a strand of DNA, right? We are a template DNA strand. Our promoter is in green. Our start point is marked right here, and we have our Tata box. We are going to have transcription factors bind to the promoter. There's a special protein called like the Tata binding protein. I'm not going to go too deep into, but there's a special protein that binds to that Tata box that's right here. We have some other transcription factors in there. When all of those transcription factors are in place, that is when RNA polymerase 2 will bind. When RNA polymerase 2 is bound, we have all of our transcription factors in place. This is called the transcription initiation complex. And once everything is in place, RNA polymerase will split that up a little bit, start our transcription, and the initiation is complete. And it will move into our second phase, which is elongation. Second part of transcription, elongation. When RNA polymerase 2 will move through DNA, it will untwist the double helix. It'll expose 10 to 20 nucleotides for pairing at once, and then it will add RNA nucleotides to that 3' end of the transcription. Goodness gracious. The RNA transcript. That's the word I'm looking for. When the RNA polymerase moves past DNA, right, as it moves, the RNA will actually peel away from the DNA. That bond isn't very strong. The RNA will peel away from the DNA, and the double helix will just reform. The hydrogen bonds will reform. It'll twist right back up to the way it wants to be. And what's cool about this, because RNA polymerase is kind of an all-in-one thing, it doesn't need other molecules to come in and prime for it. It doesn't need helicase to unzip for it, right? Many polymerase molecules can simultaneously transcribe a gene. So one RNA polymerase can start on a gene, and then before it's done, another one can get on there. If it's long enough, if the gene itself is long enough, another one can get on there. As long as there are enough transcription factors and enough RNA polymerase 2s, they will just go and go and go and go and go. So our RNA polymerase. Well, just kind of keep building and keep building and keep building until its last phase, which is termination. So, termination, the last step, happens differently in bacteria than it does in eukaryotes. So, step 3, termination, specifically in bacteria, the terminator sequence, they'll run into that terminator sequence. The RNA polymerase will detach, and then the mRNA is done. So, the transcription that you have learned in past biology classes is practically bacterial transcription. Eukaryotic transcription is a little different. So bacteria will reach the terminator. The RNA polymerase will reach that terminator sequence. The terminator sequence will be like, "Hey, stop," and then it will detach, and the mRNA will go off and do whatever the mRNA needs to do in translation, which we'll learn about on Monday. The eukaryotes, as always, are a little bit more complicated. Plenty of time. So, termination in eukaryotes is a little bit different. RNA polymerase 2 will transcribe a DNA sequence called the polyadenylation signal. Polyadenylation signal, which is AAUAAA. When it transcribes this sequence, special proteins in the nucleus will bind to that signal, will bind to that part of the transcript. And then, as it keeps going, as the RNA polymerase keeps going, Once it gets 10 to 35 nucleotides downstream from that signal, proteins will cut the RNA transcript. So that they'll hit that signal, proteins will bind to that signal, and a different protein will come through and snip off the transcript from the RNA polymerase. That transcript will then be modified, if you talk about it, after we figure out what is going on with the RNA polymerase, because it's still going. We've just released the mRNA from it. We got what we wanted. We got our transcript. It will now go be modified. But the RNA polymerase is still going. So there's another step in termination. We'll get this fancy signal. Proteins will bind, and those proteins will snip off the end. About 10 to 35 nucleotides. So we've got our pre-mRNA. It will go be modified, as we'll talk about. But our RNA polymerase is still running rampant. Our RNA polymerase hasn't detached from our DNA yet. So what happens there? Special enzymes will start to degrade the RNA strand with our new exposed end. So after our transcript is cut, after we have what we need, it's still transcribing, so special proteins are going to come munch off the ends of that RNA like the ticket machine at Chuck E. Cheese. You break it down one by one. Top them up. And this will go until those enzymes that are degrading catch up to the RNA polymerase. When they catch up, the RNA polymerase will be like, oh, okay, we're done. And it will dissociate. So our RNA polymerase will keep going. Our special enzymes will be like, hey, wait, we don't need this. We don't need this. Munch it all up. And once they catch up, they're like, hey, stop. And the RNA polymerase. So a couple more steps in bacteria, but nothing too crazy. Special munching enzymes catch up. And as they break this down, they just break them down into new nucleotides, which are free floating in the nucleus, and they will be used for the next round of transcription. Nothing too crazy. It's not like they're lost forever. Cells are really good at recycling. So, RNA polymerase is done. Until it goes back and starts a new one at the next promoter. But we have this pre-mRNA transcript. We have this piece of RNA. It's not quite what we need yet. There's a special process that we use to get that pre-mRNA to be the mRNA that we know and love and will translate to. So, just as a general overview, initiation, we've started binding. Elongation, we have our mRNA. Termination, we've released. This is our pre-mRNA transcript. This will go into mRNA processing. In mRNA processing, enzymes in the nucleus will modify that pre-mRNA in some specific ways before it's released into the cytoplasm to be translated. Special enzymes, this all happens in the nucleus. So the pre-mRNA becomes the RNA, or the pre-mRNA becomes the pre-mRNA becomes the mRNA due to special enzymes in the nucleus. So, what are those modifications? First things first, we have to do something to the ends of that RNA. We've got some pretty rough ends to it. We got to make sure that RNA is protected. So the 5' end gets a special modified guanine added to the 5' end after the transcription of the first 20 to 40 nucleotides. So after we get a decent ways down, before the end is even done, we will have this special guanine added to the end, or the beginning, I guess. That's a cap. It makes sure that nothing comes in and degrades the stuff off the beginning. At the very end, we get this poly A tail, or polyadenine tail. So after we have our AAUAAA, right, our signal, a special enzyme will come in and add 50 to 250 adenine. This is important because stuff can fall off that 3' end. And if something falls off that 3' end, we want to make sure that it's nothing important. Ah, that happened. Right. Yes. So 5' end gets a guanine cap. 3' end gets a poly A tail. Why? There are actually 3 big functions. I believe 3. If I on the next slide, right? 3 big functions that these special modified ends give to the RNA molecule. So, number 1, they help export the mature mRNA. Those special beginning and ends help it more easily slide through the nucleus to get to the endoplasmic reticulum where it will be translated. It also helps protect from the degradation of the actual transcription stuff. The special 5' cap make sure that no enzymes come through and on one side, and the poly A tail helps make sure that if something comes in and takes a bite off the end of that transcript, that it's not going to take anything important with it. It also helps the ribosomes attach to that 5' end once it reaches the cytoplasm. So that special guanine that it added to the top helps that bind to the ribosome. These caps at either end of the cell are not translated. They don't get translated in the next step. These, in addition to some other things in there, are called UTRs, or untranslated regions. So the 3' and 5' end of the mRNA have other functions, like telling the ribosome where to start, telling the ribosome where to stop. They don't get translated. There's a couple of them. So just because you have a transcript and a piece of mRNA, not all of it gets translated. There are other instructions about what to do. So this cap and tail are also are special UTRs, but there are other UTRs in the mix. So these caps and tails are not the only RNA modification that happens. There is some snipping and slicing that goes on. So there's this process called RNA splicing, where large portions of that transcript is removed, and then the remaining portions are reconnected. So a lot of eukaryotic genes have really, really long stretches of nucleotides that don't code for anything, that are kind of interspersed between the actual coding segments. So these non-coding segments, so the ones that won't end up in the final transcript, are called intervening sequences. So we call them introns. Regions that are expressed are called exons. I learned this as introns go in the trash. We don't want them. Introns go in the trash. I think my high school biology teacher took a Sharpie to a trash can and wrote introns on it. Introns go in the trash. We don't need them. Exons are expressed. That's what we want. So if we have a big long transcript, big long transcript, lotion on my hands. There's a picture on the next slide. So we have introns and exons. There are special splicing enzymes called spliceosomes, that will go through, chop up, and reconnect the important parts of our RNA. Here's our picture, right? We have our guanine cap, our adenine tail. All of our exons are highlighted in red. All of our introns are not highlighted. Um, our splicing will happen. It will chop up all of the introns, get them out of there, and glue the important parts back together. This is done by a complex of proteins and mRNAs called the spliceosome. Which is fun to say. Spliceosome. It will bind to a short nucleotide sequence in an intron. So introns will have a certain sequence that the spliceosome will bind to. The intron will be released and rapidly degraded. So it'll chop, and then put it out into the nucleus where it'll get munched up. And then it will join the two exons together. So on either side of the intron, there's an exon. It will pop them both together. This is all done by this one complex. A pre-mRNA is released from the RNA polymerase. Special enzymes will chuck that 5G cap on there and that poly A tail on there. Then our spliceosomes will come in and they will chop out the introns and glue together the exons. That is our mature mRNA. After all the exons are put back together, after all the introns are chopped up, we've got our tail, we've got our cat. That piece of RNA is ready to go out of the nucleus to the ribosome to be translated into the concomitant. A couple more things I want to hit there. Hopefully we did. So, you don't have to write this down. I just want to talk about it. There are special things called ribozymes. Ribozymes, we kind of figured out. So the spliceosome, right, had little pieces of RNA in it. Those pieces of RNA aren't only in the assembly and site recognition of those introns, they actually help catalyze the reaction. So these pieces of RNA are acting like catalysts, which is really, really cool and something we hadn't seen before. And then when we discovered ribozymes, we figured that's what the RNAs in these spliceosomes are doing. The RNA molecules function as enzymes because of three special properties. Number one, they have special 3D shape because of the single-strandedness and the sequence of molecules. They also have bases that contain functional groups that are really good at catalysis. And they have special hydrogen bonds that give it specificity. So, pieces of RNA can act like enzymes, and that is super stinking cool. We will talk about why that is evolutionarily important on Monday, right before we get into translation. Have a safe, have a warm weekend. If you need anything, let me know.